Without further ado, it is my absolute pleasure to introduce our speaker today, Dr. Sokol Todi, who is a professor in the Departments of Pharmacology and Neurology at Wayne State University and the School of Medicine, Wayne State University School of Medicine, and the chair of the Department of Pharmacology. Dr. Todi's laboratory investigates the mechanisms of neurodegeneration and neuroprotection in age-dependent neurodegenerative diseases caused by misfolded proteins, with a particular focus on polyglutamine disease proteins. His lab has investigated SCA3 for over a decade and has recently expanded to other SCAs, including SCA2, SCA7, and SCA17. The Todi laboratory uses cell culture and the fruit fly, Drosophila melanogaster, to model different spinocerebellar ataxias and other diseases to find potential therapeutic entry points that include small molecules as well as lifestyle options such as exercise. So we have a fantastic talk ahead of you today. So without further ado, I'll turn the floor over to Dr. Todi.
Thank you very much, Dr. Stewart. I am thrilled to have been invited to speak with you today about SCA2 and what's going on in the SCA2 research and development field. I will do my best to be as understanding and as understanding as possible. Sometimes, though, I do know that I talk too quickly. I'll try to slow down. My mother always told me that. Sometimes I remember, sometimes I forget, but if I need to slow down, please don't have any hesitation to interrupt me and say, Sokol, slow down, it's too fast. So I'll start with a bit of information on the biology of SCA2. I'll then talk about the epidemiology of it and model organisms that we use to study and understand this disease and to develop therapeutic options for it in the clinic. And also give you some ideas on what's going on at the clinical trials level about SCA2 right now, things that have transpired, things that are about to start, things that are ongoing. Some of the text is going to be small to read, but I will help you by giving you the most important information. And it is my understanding that after this is done today, our time together, you'll have access to the slides and the recording to read things in more detail should you want to. Of course, if you have questions that we don't cover today in the Q&A session, please feel free to email me directly. I'll be happy to communicate with you. So SCA2, it was first actually described symptomatically in India in the 1960s and the 70s, but it was officially identified in 1996 in seminal work conducted by Dr. Stefan Post, who remains a leader in the field and who has conducted a lot of studies that I will mention today in briefing. So I want to thank Dr. Post at the very beginning for being a giant in the field. In terms of how it shows in patients, SCA2 is primarily a gait ataxia. There is also muscle cramping as well as some problems with eye movement. Other symptoms of SCA2 can include neuropathy and frontal executive dysfunction. It is caused by a mutation in the gene known as ataxin-2, which has this region inside it that contains these SCAG nucleotides or repeats that, when expanded beyond a certain threshold, cause SCA2. As a dominant disease, you only need one copy of a mutant ataxin-2 to show the symptoms. This repeat usually is in 15 to 32 repeat range. Beyond 32, things become problematic, and it can go into the hundreds and become very problematic. So there is a link between severity and the age when the disease begins with the length of the repeat. The longer the repeat, the earlier the onset, the worse the disease. Based on tremendous amount of work conducted in cells, in yeast, in mice, in fruit flies, in worms, it appears that the ataxin-2 mutation causes something funny in the manner in which the protein ataxin-2 functions in the cell. So it still can perform functions that it usually performs, but also starts misbehaving by doing things it's not supposed to. What we know about the functions of this protein so far is that it's very important in the metabolism of RNA, which is the messenger between the gene and the protein. And it's such an important protein, this one, Ataxin-2, that it's also been implicated in ALS or Lou Gehrig's disease, as well as in modifying the symptomatology of other SCAs like SCAs1 and SCA3, and even in Huntington's disease. So a very critical protein with a tremendous role in the cell that, when mutated, causes problems in patients. This may be scary as a slide to look at in the beginning, but I wanted to show you a very quick diagram of how we think of Ataxin-2 as a protein. It's a pretty large protein. The expansion that causes the disease is right here at the beginning, or the N-terminal portion of it. And it has multiple kinds of domains and regions that allow it to interact with other proteins and to perform its functions. And so a lot of the work that has been done to identify these functions, again, was done in different model organisms, and I'll name those in just a moment. In terms of diagnosing SCA2, this is done through neuroimaging, electrophysiology recordings, as well as biomarker tracking, and the field of biomarker tracking is expanding. I'll mention a few of the current efforts at the clinical trial level about this later on in the presentation. Motor and cognitive assessment, of course, is also a key component of diagnosing SCA2, and of course, sequencing to see what the mutation is within the Ataxin-2 gene. The things that tend to go wrong in neurons and other cells in the nervous system because of the Ataxin-2 mutation include the aggregation, the clumping of the toxic protein into things that don't come apart very easily again. There is oxidative stress, so problems with oxygen modifying certain proteins and DNA. There is a problem with the manner in which the ion calcium is shuttled within the cell, where it is, how long it stays there, and how it's removed. And calcium is a key component of the life of the cell, and especially in neurons that are very electrically prone cells. We need to maintain a very tight regulation of where calcium is and how long it's let out of its reserves, and then how quickly it's pulled back. And that is impaired to some extent in Ataxin-2 mutations. And there is impairment with the way in which DNA is processed, meaning how DNA is used to generate proteins from its message. The age of onset depends on the length of the CAG repeat. As I said before, the longer the repeat, the earlier the onset. The average age of onset is about 35 years. A longer disease duration does correlate with greater disease severity, worse balance, and a higher functional dependency overall. Now, SCA2 is the most prevalent of the polyglutamine ataxias, and these include ataxias 1, 2, 3, 6, 7, and 17. So it's the most prevalent among these ataxias in Cuba, India, Mexico, and Southern Italy. The largest prevalence is in Cuba due to a prominent founder effect, meaning populations that were started by a group of people who carried this mutation and were able to transmit it over next generations. And the frequency there is about 183 carriers per 100,000 inhabitants. The SCA2 survival rate, as well as its prevalence, does depend on the geographical location and ethnical groups. So, for example, the longest survival is seen in Italy with about 67 years, whereas the shortest one is in Spanish and Cuban groups with a range of 52 to 54 years. How do we study this disease? We know the mutation, and we understand a little bit on how the protein functions, and we want to use this information to come up with, therapies that can be beneficial in the clinic. But what do we use to gather this information and to try and translate it into therapeutics? Well, a lot of different systems have been used and continue to be used for SCA2 to understand it These include cultured human cells, cells that were isolated from people a long time ago, like the HeLa or the Henrietta-Lacks cells that are kept in labs in perpetuity. Scientists like me would introduce a mutant version of Ataxin-2 in these cells, and we would look at how the protein is functioning. Is it doing the same thing as the normal version, or is it doing something different? Is it going to a different place in the cell? What sort of things are happening in the cell because of its presence? Things of that nature. Most recently, scientists in the field are using what are called iPSCs, or induced pluripotent stem cells. These are cells that are actually donated by patients who are afflicted by SCA2, and these cells now we can differentiate or make into almost any cell we want in the lab, whether a neuron, a muscle, a glial cell, you name it, to understand more the function of this toxic protein ultimately in the natural environment. But these are still cells in a petri dish, often just as a single layer, sometimes in multiple layers, and they don't tend to tell us. They're very important to understand what's happening at the cell level, but to understand at the organism level, we use then model organisms, and these can include yeast, this can include the worm C. elegans, these can include the fruit fly, Drosophila melanogaster, and I'll show you an example of how we use flies in this case. And of course, the ever-present, ever-useful, ever-important mouse models of SCA2, of which there are a few that have been generated that have been very, very important. And so we use different models to put together sort of the pieces of the puzzle on why the protein is toxic, when does it become toxic, and what can we do to it or the cell to either reduce or prevent toxicity. And once we learn this from cells, we then can ask these questions in an animal, say, can we do this in a mouse? And does it have the same benefit that it has in the cell? And then there are things that you cannot do to a cell that you can do in a model, like exercise. I mean, we could try to come up with an exercise paradigm for cells. I don't think it's going to work very well, but we can use organisms, full intact organisms for that. And in fact, I'll give you an example of this that we published a year ago about the utility of using animal models and exercise to understand the benefits of exercise in different diseases, including SCA2. So I'm going to show you a movie. And I want to just look at it for a moment. In a few seconds, a hand is going to appear and is going to force the flies to the bottom. And you'll see they move up. Some of them, these 3 vials, move better than these other 2. Now, this is a normal behavior of the fly, this negative geotaxis effect, meaning moving against gravity. And we use that to this effect, and we can actually numerically assess it very carefully and reproducibly with a lot of scientific rigor to understand the impact of different disease proteins like Ataxin-2 to the longevity and the motility of the fly. Now, because of this negative geotaxis effect, that means that we can actually design a machine to exercise flies. You saw how when Allison's hand kept them down to the bottom, they moved up. Well, if you keep doing that over and over and over again for several hours each day, for a number of days, you can actually exercise flies. And when you do this with just normal flies that you can even pluck outside where your bananas are hanging or somewhere else, they will actually slow down their aging, and they'll perform better in mobility and motility long term. So a few years ago, I was at an National Ataxia Foundation investigators meeting, and people were talking about therapeutic benefits from different things that we can do, and the possibility of exercise having an impact as a therapeutic option of some sort in ataxias generally came up. And so when I came back to my lab, I talked to a collaborator of mine whose office is exactly downstairs from mine, just the floor below. His name is RJ Wessels. I said, RJ, can we see if we can check if exercise can be helpful for polyglutamine ataxias, and this included SCA2. And so what we did is we took flies that expressed the mutant protein of ataxin 2. And then we expressed it in fly neurons, in all fly neurons, or in all tissues, and we exercised them. And of course, then there were the siblings that also expressed ataxin 2 that did not exercise. And so now I'm going to tell you what we found. So these flies right here on the left are flies that do not express any mutant ataxin 2 that have been exercised. They move very briskly. I think we can agree on that. These flies here on the right, these two vials, are flies that express ataxin 2 that have not been exercised. And these ones, I think you see where I'm going with this, these ones right here in the middle, are their siblings that express ataxin 2, but they have been exercised. And we found that exercise led to longer life, improved mobility, and lower levels of this toxic ataxin 2 protein in the fly. And so exercise seems to have a benefit in this direction. We did a lot of work to understand how, and we have a few things in motion right now on how that happens. I'm happy to address them at the question and answer session if you're interested, or I can tell you more by email without getting too technical. And this idea of exercise being important, potentially in SCA2, is actually supported by evidence from various studies conducted with patients suffering from SCA2 or other types of ataxia. I know this is small, so I'll just tell you what was found. So a study conducted or published in 2019, so just a few years ago, looked at SCA2 specifically and neurorehabilitation over the course of 12 weeks. And what they found is improved overall balance, improved finger to nose touch, heel to shin touch, and a reduced time in terms of crossing a 5-meter path. So exercise improved things overall. This was confirmed by studies conducted in other ataxias, including Friedreich ataxia, SCA1, 2, and 3, SCA7 as well. And overall, they used different modalities of exercise. But in every single item tested afterwards as an outcome, things were improved, either a little or quite a bit in terms of balance, in terms of para, that sort of modality of speech, and other things, cadence, gait parameters, and whatnot. So overall, exercise for these diseases seems to be beneficial. I will caution here that there are diseases where exercise makes things worse. In published work, that has been the case for the Kennedy's disease, for example, but for SCA2 and everything that's been done so far, exercise has been deemed beneficial. So we use different model systems to study and to understand ataxin 2 and SCA2. The first model systems at the organism level were the yeast, the worms, and the flies. They allowed for the identification and understanding of basic functions of the ataxin 2 protein. The mouse models then enabled scientists to further expand on these findings and to test therapeutic possibilities. Several models of SCA2 have been generated in mice and have helped us to understand the biology of the disease, as well as devise options for therapeutic intervention in the clinic. Now, what are some of the things that we have used these mouse models for? Well, basic research. What is the function of ataxin 2 in a cell? Electrophysiology. How do the neurons behave electrically? What are the motor anomalies that we see when ataxin 2 is present? And these are all very important modalities because then we can check on their improvement or not, and in some cases even worsening, when different types of therapeutic options are pursued in these models. But ultimately, they're not humans, as we know. They age very differently than us. And there are key differences that can't be recapitulated in models, generally speaking, in mice compared to the human patient to help us understand these diseases, including SCA2, more in the native environment. That's why scientists have started using more and more the induced pluripotent stem cells, or iPSCs, that are derived from SCA2 patients to understand, again, how these cells are behaving when plucked out of their natural environment, and to help with therapeutic approaches as well. What are the therapeutic approaches? Well, multiple things have been thought about for SCA2, as in other diseases. I thank here Dr. Egonova and Dr. Besprosani for a beautiful research paper they published not too long ago about SCA2 and different therapeutic options. I am borrowing this slide, this image from that manuscript. A go-to therapeutic option recently for SCA2 and other diseases of the same family have been antisense oligonucleotide, or ASOs or ASOs. So what do? Well, the idea here is to prevent the protein, the toxic protein, from even being produced. So if this is the gene that encodes the ataxin-2 protein, this DNA is made into an mRNA, messenger RNA, that will be translated into the toxic protein, and ASOs can bind specifically this mRNA and lead to its destruction. Therefore, you reduce the levels of ataxin-2 protein by not even allowing it to be produced in the first place in the cell. Over 150 different molecules were tested in cells, and one showed a lot of promise in reducing the levels of mutant ataxin-2. It has performed very well in mouse models of SCA2 in terms of electrophysiology, calcium dysregulation, motor anomalies, and longevity and whatnot. The issue here is that delivery to the brain is very difficult, and there are new formulations to try and enable them to cross the blood-brain barrier. That's what the BBB stands for here, not Better Business Bureau, blood-brain barrier. And so there are efforts right now from different institutions and companies to improve the deliverability of these molecules through the brain to prevent this protein from being produced. Another possibility are stem cells, and I'm sure you've heard about this before. The possibility has arisen that mesenchymal stem cells can be injected into a person suffering from SCA2. These cells can differentiate into different things, including neurons, and the hope is that they'll replace neurons that are dying or have already died. Intravenous delivery of these cells did improve different phenotypes in the SCA2 mice, but there hasn't been a study that I'm aware of that's published the efficacy or any aspects of this type of intervention in SCA2 patients. There was a study conducted in SCA3 with only 6 patients tested. The procedure was well tolerated. Not much of an improvement was observed that was a very small study to really be able to capture minor improvements. So a larger study may need to be conducted. And in fact, as I'll speak later on, there are efforts underway to try the mesenchymal stem cells as a therapeutic option in SCA2. And I'll cover that later on. Another possibility to use for therapeutics is to prevent calcium, that calcium ion that is misbehaving. Essentially, it's mislocalized, different concentrations where they shouldn't be in the cell, is to stabilize it, to make it more normal. And because, as I said earlier, disturbed calcium signaling and concentrations are a shared feature of SCAs, and they are present in SCA2. There is evidence that regulating the calcium levels in a mouse models of SCA2 has helped improve phenotypes. The same thing has been seen in patient-derived cells. Ataxia patient study with a channel blocker, riluzole, which is also used in ALS, did improve some parameters, but that study was not specific to just SCA2. It included various SCA patients. And as you'll see, when we go to the trials information that I'll present to you, riluzole and similar other molecules that help with blocking channel dysfunction for calcium movement are still a primary focus as therapeutic opportunities for SCA2. Another thing that people are trying, and this doesn't mean that every single technique needs to be done in separation. There is possibility of multiple techniques to be used to achieve a goal here. Another possibility is using anti-aggregation. So as I said earlier, and as you, I'm sure, all know, ataxin 2, that's mutant, it's a sticky protein, it clumps. So maybe we can prevent it from clumping, and that may be less toxic because there is evidence from various studies and various models of SCA2 that preventing certain types of aggregations from happening can be beneficial. There aren't any specific reports presently on preclinical studies, meaning in animal models, to describe the effects of molecular chaperones, things that keep ataxin from clumping, but it is an area that is currently under investigation in some parts of the world, including in Europe. So these are some therapeutic options. They don't need to be the only ones. And again, they might be usable in combination depending on their toxicity and tolerance profiles. But what is going on and what has been going on recently at the state of research and clinical trials for SCA2? This information came from this source, the clinicaltrials.gov. So here we start with things that have been completed. I know it's small font, but I'll highlight the important things here myself. Some of the studies were done to find therapeutic options. Others were more about finding biomarkers that can help with studying specific therapeutic options. In other words, if somebody is undergoing a therapeutic option to try and prevent or completely heal SCA2 or slow down its progression, we want to know, is it working? And the easier the way to find out that it's working or not, the better it is. If this had to be done through MRI scanners, well, that's not very easy because they're not very easy to find. They're not everywhere. They're expensive. They take a long time. But if we can isolate things from the blood that tell us that this disease is now moving in the right direction, a toxic component has gone down or a beneficial component has gone up in blood, that's much more easily addressable. It is easy to withdraw blood, have it tested, and understand how things are working. Again, those biomarkers are very helpful in this direction. And so one thing that has been conducted so far that led to no benefits was a study conducted at the University of Florida. This included SCAs 1, 2, 3, and 6. They didn't see any benefits. And these used a potassium channel blocker, which is different than calcium. Not a calcium, it's a potassium channel blocker to look at it in terms of gait improvement. Why did they use a potassium channel blocker? Well, potassium is another key ion in the electrical profile of the neuron. By blocking it, you can try to restore some of the electrical properties that may have gone haywire in the disease. But this potassium channel blocker did not lead to any detectable benefits in this study, which was completed in 2015. There was a study conducted by BioSCA, which was to identify biomarkers in autosomal dominant cerebellar ataxias, and this included SCAs 1, 2, 3, and 7. This study showed that volumetry outperformed clinical scores in these diseases, and then they saw that neurofibrillar levels at baseline in the SA expansion carriers predict cerebellar volume change and clinical score progression. So found something that can be isolated from blood that can give you ideas on how things are progressing, regressing, or nothing is changing for these diseases. Another trial was conducted with riluzole, which we talked about before, that calcium blocker that has also been used for ALS or Lou Gehrig's disease. This study was done in France, and there was no improvement with this specific formulation. However, as somebody who chairs the Department of Pharmacology, I can tell you that the type of formulation matters a lot because it means how the drug is going to be accessible to the neuron and the rest of the brain, and how potent or not it's going to be to bind to a specific channel or multiple channels. Other studies that were conducted in 2020 and 2022, or rather completed in 2020 and 2022, had to do with rehabilitative trials with cerebrospinal stimulation. This is a transcranial direct current stimulation It's a non-invasive technique. It has been shown to modulate cerebellar excitability and improve symptoms in patients. They use this for ataxias in general, including SCA2, and a 2-week treatment with this TDCS reduced symptoms in patients with ataxia and restored some motor cortex inhibition, meaning it allowed the different parts of the brain to communicate better with each other for improved overall performance. Another one has been a study to develop and validate analytical and clinical non-invasive prenatal diagnosis for triplet repeat diseases, including here SCA2. The methods that were used here were not sadly very conclusive, so better methods need to be utilized, but this was essentially to understand how can we be better at identifying these diseases and their evasiveness prenatally. Another one that was completed, but the results are not yet available, comes also from France. France, as you can see, has been a leader in these efforts. This was the integrated functional evaluation of the cerebellum, or CERMO. One of the main objectives of this study was to validate potential biological, clinical, and/or imaging biomarkers in SCA patients. This included SCA2 and SCA7. This is for the reasons of testing downstream the antisense oligonucleotides that we talked about earlier that would hopefully prevent the production of ataxin 2. So setting the stage essentially to know what do we look for in patients to find out as non-invasively as possible if they are improving from the delivery of these antisense nucleotides. Fingers crossed for good results from this study. I'm sure they'll be available soon. Now, there are some active trials that are not recruiting. One of them is riluzole, which is a prodrug form of riluzole, meaning it becomes riluzole once it's introduced into the patient. This is still inactive. It's not recruiting. We don't know what's going to happen yet. Fingers crossed again for good results. This is from Biohaven Pharmaceuticals. And the target are SCAs more generally, not just SCA2. And this applies to the other two studies that are active but not recruiting, meaning that they are for SCAs in general, not just for SCA2, but include SCA2 patients. This is the Instrumental Data Exchange for Ataxia Study, or IDEA. The idea here is to study testing of body-worn sensors to measure movement during simple tests of coordination so that they can help clinicians with evaluation and progression of severity of ataxia, and can also be used to see whether specific regimens or treatments are having any beneficial effect or not. Another one is the open pilot trial of V4157. This is another prodrug of riluzole. This is done by UCLA, and we'll see how things happen here. The primary target is a channel blocker, is the calcium channel, but there are also some other channels that can be blocked by riluzole, so we'll see how that works out. And finally, there is an ongoing trial. This is a prospective study of individuals at risk for SCA1, 2, 3, 6, and 7, RISCA. This is to perform a prospective observational study of individuals at risk for the most common SCAs. It's ongoing. This is also being done by France, and we don't quite know yet where the study is going to go. Hopefully with additional ways that we can, again, identify the risk of individuals for SCAs, both to evaluate and diagnose them more easily, as well as track their progression as things unfold. There are two trials that are recruiting. One of them is from the Teachers College at Columbia University, the other one from University of Florida. These are also both SCA in general, not just SCA2. One of them has to do with priming motor learning through exercise in people with spinocerebellar ataxia. So the idea is to test the feasibility and efficacy of an 8-week telehealth intervention of high-intensity aerobic exercise in patients suffering from SCAs. This is, so essentially checking again exercise and its benefits, specific types of exercise. And the other one is a natural history study of genetic modifiers in SCAs. The idea here is, how does disease progress over time? What are the best ways to measure progression? And are there specific genes other than the one that's causing the disease? So are there things in the genome besides ataxin-2 that causes SCA2 that can help make things better, or maybe they make them worse overall, and that will give us more ideas on how to target things for therapeutic purposes. So calcium and other channel blockers, one target, exercise, and a lot of work being done to understand better and to be as non-invasive as possible to understand better, again, the manner in which patients present, to diagnose them more quickly, and to hopefully have all the tools we need at our hands to find out, without having expensive tests or tests that are hard to conduct, or that can only be conducted in certain places, to understand how people are progressing when certain therapies are being designed, tested, and then hopefully in the very near future administered in the clinic for patients. Finally, we have the umbilical cord mesenchymal stem cell study. This is not recruiting yet, but it includes SCA2 patients. This is Sinao Biotechnology Corporation, and this is to verify the safety and efficacy of human umbilical cord mesenchymal cells, like we talked about before for SCA3, and their introduction through intravenous or intrathecal ways. Hopefully intravenous will work very well so that it's less invasive, to see how they may help for SCA patients in general, including SCA2. That is the end of my presentation. I am happy to exit screen sharing if it helps, and we can perhaps talk about questions that may have come over.
Wonderful. Thank you so much for a fantastic presentation. And just a couple of reminders for our attendees. If you do have any questions, you can drop that into the Q&A window at the bottom of your screen. Also, one of our staff members just dropped a link in the chat to the webpage on the National Ataxia Foundation website, where you can find the latest information about how to participate in research studies across the USA and internationally as well. So our first question today is about inheritance patterns. So if someone has SCA2, do they need to have the mutation from just one parent, or does it need to have, both parents need to have SCA2 for a kid to potentially get the disease?
Right, that's an excellent question. SCA2 is classified as dominant inheritance, meaning that a single copy from only one of the parents, if it's passed on to the child, it will be problematic. So if just one parent has it, there is a 50-50 chance they'll pass it on to their children. I hope that helps it. But Celeste, you are an expert in this as well, so feel free to expand on my answer. I have no problem with that.
No, that's fantastic. We also have another question relating to the DNA and repeats. So we have someone asking here, you mentioned that the more repeats that someone has in their gene, the earlier they start experiencing symptoms. How accurately can we predict that? Or is it more of a general prediction and not very specific? Could you elaborate on that a little bit?
That's another great question. And I would say that's a general prediction rather than a very specific formula that can be used. And in part it is because, as I mentioned briefly earlier on in the presentation, the toxicity and the longevity after the symptoms appear depends on racial backgrounds, with Southern Italians doing better than, say, Spaniards or Cubans who have SCA2. And so it's a bit of a fuzzy zone there. I wish I could give you a formula, but ultimately it comes down to the longer the repeat, the better the higher the chances the disease will show up earlier and be worse.
That's really good to know. We have another question relating to life expectancy in SCA2. Is there anything known about how SCA2 impacts life expectancy, or anything relating to that?
Right. So it does impact life expectancy. So there is reduced longevity unfortunately, in SCA2 patients. And this again will also depend on the genetic background overall and where people are from. So about maybe 67 years old is the average longevity of SCA2 patients from Italy compared to 50 to 55, for example, or somewhere in there from other populations. So yes, unfortunately, it does reduce longevity. There are some SCAs that don't necessarily impact the overall lifespan. They impact more so motor abilities like SCA6 or SCA6 would be the way, but for SCA2, we do have some reduced longevity, unfortunately.
So it sounds like there's a lot of factors that go into figuring these things out.
And that's a great help to me, Celeste, to say that this is precisely one of the goals of one of the studies that are currently underway to understand how the rest of the genes may or may not regulate the toxicity of this mutant ataxin-2. And as big as the genome is, and as many things as go into changing or impacting somebody's life, including lifestyle choices, place of residence, socioeconomic status, and all of that, a lot of factors involved. The only thing we can say for certain is that if somebody has a mutant version of ataxin-2, they are going to experience some sort of symptom or another, depending on how long that repeat is.
Speaking about repeats, we have a question about genetic anticipation. Could you explain a little bit more about what that means and how much is the repeat increase between generations, if something's known about that?
To my knowledge, and I wouldn't put a lot of stock in that part personally, so I think it's worth doing some more digging, and I'm happy to dig with whomever asked the question. The precise expansion from generation to generation is not clear. Depending on the polyglutamine disease, sometimes it seems to be that if it's passed from the paternal line, it expands more so than the maternal line. But that's what anticipation is, right? The likelihood that the repeat will expand as it goes from generation 1 to 2 to 3 and so on. And sometimes it can be a short or no expansion. In some cases, it can be a pretty dramatic expansion, but there is no real fast and quick formula to determine that for polyglutamine disease in general, including SCA2 or SCA2. 100%. And I'm sorry if most people like saying it's SCA2, I tend to go back and forth between SCA and SCA, depending. So
there's different preferences.
Yes, well, there are some feelings about that in the field.
Yes, there are. So shifting a little bit to now pharmacodynamics. So you mentioned that the blood-brain barrier poses an issue when designing therapeutics. Could you expand a little bit on that and explain what different companies can do in order to make it so that it's not as much of an issue when developing therapeutics for SCA2?
Yeah, so the brain is extremely well guarded from the rest of the body in terms of access to things floating in the blood. And we don't fully understand all the mechanisms on how this works, but if you think of cells that look like hands, they will wrap around the neuron and they'll either allow more access or less access to the blood flowing around. In the brain, these cells are very, very tight, so that things are filtered very carefully on what the neurons are exposed to, including also glial cells residing in the brain. So that's a blood-brain barrier. You have a tight grip around it. Things have to go through cells instead of in between them to enter the nervous system. What can we do for that? Well, one might say, hey, let's reduce a blood-brain barrier. I would caution against that because other things that we don't want, including lots of pathogens that may be in the blood, maybe, or other things, can then come into the brain and cause a lot of other troubles. But what can be done is to generate compounds that are friendly to membranes, meaning they can enter a membrane and exit it easily so that they enter a cell, stay in the cell for a while, and then they exit and are available to the neurons for uptake and to the glia for uptake. So if we make things that are more membrane friendly, it's great. Or or if we make compounds that have specific receptors or channels to which they can have access in the cells that make this blood-brain barrier, that will also be helpful. But we shouldn't expect something that's in the blood to simply just diffuse, go to the brain. There's going to be a barrier there keeping it from entering. I hope that helps.
Totally. So it sounds like the blood-brain barrier is kind of like a border control crossing, and you need to have the right passport to get across easy.
Yes. And there are only certain gates you can go through.
That just makes things very complicated. We have another question about the brain. So what does the cerebellum look like for someone who has SCA2? And is there anything known about how the cerebellum might impact issues with the eyes as well?
Right. So the cerebellum, which is key in coordination of body movements and actually provides help with some cognitive functions as well, is impacted in SCA2. There is loss of the cerebellar volume. So that means that we can't coordinate things as well. Things that shouldn't be happening are happening, and things that should be happening aren't happening as easily anymore. And that will impact to some extent the eye movement as well. But it's not the primary reason for the issues of eye movements. There are other centers in the brain that control eye movement as well, that coordinate through the cerebellum, as I say, cerebellar cortex. So, but yes, the cerebellum is impacted. It does play some role in the eye motility issues, but it's not the primary culprit for that part. Other centers in the brain that are motor related will be impacted as well.
And then we're going to shift gears a little bit here in terms of question topic. You talked a lot about exercise during your presentation. We have a number of folks who have questions about what specific kinds of exercise benefits SCA2, and if there's anything we know about what you're testing out in the flies and how that translates to human patients.
So yeah, that's absolutely fantastic. I mean, it's known that exercise is important for almost any modality you test. And in terms of SCA2 specifically, or SCAs more generally, it seems like almost any type of exercise activity is helpful. Yes, there can be high intensity. There can also be medium or low intensity. But from the studies that have been done so far, overall, there is help from exercising. However, there's always a however. Based on, and this doesn't apply to SCA2, but it's something to keep in mind more generally. Yeah, there could be some cases where intense exercise can be problematic. This appears to be the case with ALS. This seems to be the case with Kennedy's disease. So for these diseases, for example, in my lab, we are testing different kinds of modalities for exercise, and we have found that for the Kennedy's disease, high intensity is bad. They do really, really badly with that. But if you keep them at a lower intensity, gentler exercise, long-term, they do much, much, much better. But for SCA2, everything that we know so far, and there is clearly more work being done, especially now to understand the potential benefits of high intensity, it seems that exercise in general, whatever you can do that's possible, sounds great. I'll give you one more thing though. I'm not involved in this study. One of my collaborators is, but there is a group testing the possibility that simply having the idea of exercise to virtual reality, whether that may alone stimulate some improvement. That study is still in the planning phases, so it's still very, very early, but it has been approved for funding. And so we'll have some ideas from that as well, that maybe wearing some exercise goggles gives you a notion that you are doing something and tricking the brain into thinking that you are exercising. We'll see.
We'll have to keep an eye out for that study. We have another question here, and I think this one's actually for me. What testing can my offspring do, or what I do to test out if we have the gene for SCA2? So we actually have an information page available on the National Ataxia website, and we'll have someone drop that link in the chat here. We actually have a
Perfect.
free genetic testing program for folks with SCA2 who are based in the USA. And if you are based outside of the US, we also have links to other companies where you can find out information on how to get tested for SCA2. This can be helpful so that you can participate in the research studies that were previously mentioned by Dr. Todi. Oftentimes they require genetic diagnosis in order to participate in those studies. So genetic testing is a great first step. And there's also options for different family planning options after you receive that genetic information. Then our next question, Dr. Todi, could you explain a little bit more about the gene editing options that are being in development for SCA2 treatment?
So there are some talks and some studies thinking about the possibility of using something like CRISPR to edit things out, and in other cases in, but in SCA2 out, so to reduce essentially the length of the ataxin-2 gene from a long repeat to a shorter repeat. That's still very early on in terms of studies. And part of it is because it is not an easy thing to do to restrict just a specific, very repetitive sequence this way and to cover the entire brain. So there is one thing. I suppose we could think of antisense oligonucleotides as gene editing as well to some extent, because they're targeting a outcome of a gene's production with the mRNAs. So there is work, as far as I know, going on with that. But I think that somebody from the Ataxia Foundation is even better versed than I am in terms of what companies are doing with ASOs for SCA2. So those are all interesting and good and highly promising. But I think the field right now, especially in terms of directed gene editing, is not quite where we need to be to see it for prime time implementation overall. We have a lot of things to still figure out in terms of, well, how faithful is this gene editing going to be? Is it going to only target the ataxin-2 gene, but might it have some sort of other things clicking and moving in other parts? And then once it's introduced into a patient, will be able to reach every cell it needs to reach, only some cells? And how do you get it there with the blood-brain barrier issue as well? I mean, that could be bypassed in some ways by injecting directly into the brain if it's a one-time thing. But if you have to do it over and over and over again, becomes more complicated.
So these very exciting technology, but still a lot more work needs to be done in terms of safety and understanding how to best try this out before we're going into humans. That's why we are going to need those animal models that you were talking about to make sure things are working well.
Yeah, and I mean, that's the thing about working with the brain as well. It's such a complex organ. We still know, or we still don't know a lot about it. And so translating these possibilities that can be much more easily used for other organs, translating them to the brain takes quite a few more steps.
And then on the topic of the brain, we have a question here. Is there anything known about a link between SCA2 and different mental health problems?
That's a very good question. And by that, I mean that I am not certain. So I'll have to look into that and get back to you. I mean, SCA2 patients do show some frontal cortex issues, some cognitive impairment, but I am not certain how prevalent that is. And so I cannot speak to that, unfortunately.
Oh, no worries at all. I can elaborate on this a little bit from sort of the patient support side of things. It can be very difficult when you receive information about a genetic diagnosis relating to neurodegenerative disease. We have a lot of folks who've reached out to us looking for support and comfort and are not quite sure how to process this. So if you or someone in your family is struggling, that is very, very normal. And there is support out there. So depending on who you are working with in terms of your primary care physician, they may be able to put you in touch with different mental health specialists who are familiar with ataxia patients. And there's also support available through our support groups at the National Ataxia Foundation, because oftentimes chatting with someone who understands what's going on and has similar life experience and having that solidarity can make all of the difference. And then just going through, we have about 3 minutes left, and we'll try to get to as many questions as possible. We have a question here. A lot of the studies that you shared and talked about that are currently in clinical trials were on adults. Is there any work being done for on the pediatric side of things? Do we tend to see SCA2 cases in children?
That's a great question. I am not aware of any studies going on with SCA2 in children. SCA2 is not a disease that has really prevalence in children. It's at a fourth and following decade of life. So that's probably the reason why there isn't much work or any work, to my knowledge, being done in that direction. Unlike some other SCAs or SCAs that with very, very, very long extensions can show up very early on. This one is not the case there.
Mm. And then we have a question. You mentioned in your presentation that there was a drug called riluzole, and then they formatted it in a different way to create a slightly different version of the drug called troriluzole. Could you expand a little bit more about how they did that, why they did that? Is there a way they could do that to make it better for SCA2 specifically compared to other kinds of ataxia?
Right. So the reason for having these different forms available is to just, again, make them available as easily as possible to the brain. And often, prodrugs will work better than just a drug in crossing that blood-brain barrier and being available to the actual cells that require it. Now, in terms of specificity, riluzole is called as a channel blocker, as a calcium channel blocker, but it does target other channels as well. We do know that there is calcium dysregulation in SCA2. So what if we were to modify riluzole into something that's more specific for calcium? Might that be more beneficial to SCA2 patients than others? I don't know, but we'll have to see from the specific outcomes of these trials, is there any benefit at all? And if there is a benefit, which SCAs or SCAs are benefiting the most? And once we have that information, then we can start seeing, well, what were things that were improved in SCA2 compared to SCA3 compared to SCA7, and what things weren't? And how can we use this information now to see how we can target one better than the other and make different formulations from these first generation, shall we call it, drug into improving it for specific ataxia. So yeah, there is a possibility there.
Well, thank you for a fantastic explanation. We are at time, so if everyone could join me at your own computer, thanking Dr. Todi for joining us today and giving a fantastic presentation on some of the research that's being done on SCA2. And thank you all for joining us today to listen to this session and learn. I hope you have a wonderful rest of your day. Thank you again for joining us, and hope you have a wonderful rest of your day, wherever you are joining us from the world.
Thank you.